Fellow 90s kids, and non-90s kids, do you recognize this? If you're like me, you probably took the pink bubblegum medicine at least once as a kid. This is amoxicillin, a form of the antibiotic penicillin. Antibiotics are drugs that specifically target bacteria, so you probably took this to fight or prevent a bacterial infection. But bacteria aren't the only things our bodies need to fight off. Unless you're super naturally healthy, you've probably gotten a cold a few times and maybe the flu. But these illnesses are different from bacterial infections. They're caused by viruses. And these pathogens keep changing and evolving. In fact, there are over 100 known viruses that cause the common cold. We don't commonly use any medicines akin to antibiotics for viruses like the common cold. And yet your body's immune system manages to fight off any number of these different viruses, along with tons of bacteria and other pathogens on a daily basis. How exactly does the immune system stay on the grind like that? Let's start with how the immune system broadly works. Our bodies fight off disease agents in two ways, with the innate immune response and the adaptive immune response. Innate immunity refers to what your body knows to defend against already. This can include cells that destroy and consume infected cells, or general barriers to infection, like your skin or mucous membranes. Adaptive immunity gets more specific. It involves targeting an invader directly, once the body sees it, through processes like making antibodies against them. This part of the immune system is how vaccines work. Your body doesn't recognize that pathogen yet, so the vaccine shows the adaptive immune system what to look for. While there are a lot of different types of cells involved in adaptive immunity, they all rely to some extent on antibodies, which are made and secreted by B cells to recognize foreign proteins or antigens. For the rest of the video, we will focus on a pillar of the adaptive immune system, our body's extensive libraries of antibodies. The antigens that antibodies bind to and recognize as foreign can be proteins on the surface of viruses or bacteria. When an antibody binds an antigen, it marks the invader or an already infected cell for destruction. Let's take a closer look at antibodies, the powerhouse of the immune system. They also have a fancier name, immunoglobulin, and are made up of two protein components, a heavy chain and a light chain. The parts of the antibody that recognize antigens are here, at the ends of the Y shape. This region of the protein has specific properties that let it bind to the antigen that this antibody was made for. Broadly, there are 5 classes of antibodies made by B cells that are able to specialize in different immune tasks. But let's hear more from an immune system expert.
As a B cell goes through development, IgM is the first immunoglobulin to be expressed. In fact, most B cells express both IgM and IgD by the time they're mature. This all happens while the cell is still naive, or before it's ever seen any foreign antigens. Think of IgM and IgD as security guards that will investigate anyone suspicious, even if they're not necessarily on the wanted list yet. The difference between them is that while IgD tends to stay at the security booth, IgM is bigger, buffer, and can be dispatched or secreted outside of the building to chase down any invaders once the alarms are going off. Now, let's say you've had some thefts and you know exactly who's breaking in. So your guards, the B cells, are now experts on what the thieves look like and the tactics that they use. The guards can now specialize by making different classes of antibodies, IgG, IgA, and IgE. IgG is smaller than IgM, so it can fit into tighter spaces. It can also do some neat things like poke holes into membranes and alert cells that specialize in eating and destroying antigens that, hey, this is a bad guy. When the thieves try to strike again, these guards are the ones coming out in full force. IgG is most common in blood and extracellular spaces, while IgA is the main antibody in the secretions of your intestinal and respiratory tracts. It's also secreted in breast milk to protect newborn babies. IgE was the last class of antibody discovered, and it usually hangs out on the surface of cells, ready to respond quickly to parasites and allergens.
Each class of antibody has a lot of structural differences that let it do its specific job. But in order to bind a specific antigen effectively, the antibody needs to have a pretty particular set of traits. That is, structural properties of its heavy and light chains that make it especially fond of its antigen. So the immune system basically needs to be able to generate custom-made antibodies and produce a different antibody for all the numerous antigens our bodies encounter every single day. Seems like the immune system really has its work cut out for it. One way it distributes the workload is by assigning one specific antigen to a B cell, so each B cell can focus on producing one type of antibody once it matures. Well, okay, great division of labor, immune system. If every B cell is vigilantly watching for one specific antigen, with billions of cells in your body, we've got this on lock. But the hard part is training each B cell to be able to adequately recognize and bind its specific antigen, and figuring out how to produce a huge range of diverse antibodies in the first place. This process happens mainly during B cell development, and draws inspiration from evolution. To understand how this variety emerges, we have to look at what's happening at the DNA level. Proteins, like the ones making up antibodies, come from mRNA, which comes from DNA. So the variety in antigen binding regions originates at the DNA level. Any changes to the DNA reflect in the antibody as different antigen binding regions. During B cell development, the immune system uses this to its advantage with a tactic called VDJ recombination.
As a B cell develops, every cell begins with the same DNA. This DNA contains many variations of segments called V, D, and J segments, which stands for variable, diversity, and joining. VDJ recombination involves taking one copy of each segment and linking them together to form the final sequence. The V and D segments provide most of the variety. The J segment connects them with the rest of the antibody. Just rearranging the segments can give millions of possibilities for different binding sites, but as the segments are linked, the recombination enzymes can accidentally insert or delete nucleotides, creating even more chances for diversity. The final product is called the variable region, and it does most of the work in creating the part of the antibody that binds to the antigen. Altogether, this process can create 100 billion possible sequences.
VDJ recombination comes with a risk, though. What if the randomly chosen segments combine in a way that binds to your own proteins? This could lead to an autoimmune disease. To make sure this doesn't happen, the newly formed immunoglobulin is tested. And if it binds self-proteins, it tries again and can keep trying other segments until it finds one that doesn't bind to your own proteins. So the immune system also uses VDJ recombination to act as a sort of boot camp, making sure that B cells are trained to recognize only their enemies, meaning that they're not self-reactive. The immune system supervises B cell and antibody maturation to teach the cells what the other employees or cells look like, so an antigen will only be bound by the B cell if two things are true. First, the B cell gets a strong notification of an intruder antigen, like a loud noise heard from the security desk. And second, the antigen is not recognized as part of the self. Only then will the B cell and its antibody guards kick into gear. But once B cells are activated or given their assigned antigen, they need to get more specific and fine-tune how strongly their antibodies bind to the antigen. This not only lets a parent B cell bind to its antigen better, but also helps make more diverse daughter B cells that still maintain a similar target. To do this, on top of VDJ recombination, B cells undergo a process called somatic hypermutation. In this process, the recombined sections of antibody DNA are mutated even more at the level of nucleotide bases. And the hyper in hypermutation is no joke. Compared to other cells in the body, B cells can experience a mutation rate 10 million times higher. This puts the potential for different outcomes way over the top. From just VDJ recombination, we could already make tons of antibodies just by rearranging gene segments. So adding in somatic hypermutation makes the space of possible antibodies nearly endless. With recombination and hypermutation in place, a dividing B cell can yield a huge variety of antibody genes over several generations. And the more antibodies we have to choose from, the more likely we are to find the perfect fit for an antigen. Remember how I said earlier this process is like evolution? Well, the search for the right antibody over many iterations of B cell development happens to look a lot like evolution. But in your immune system, the evolution occurs over generations of cells, instead of over a population of organisms. Evolving an arsenal of diverse antibodies to recognize all sorts of potential invaders is no easy task, but your B cells do it quite well. Thanks to them, your blood has more than 100 million different types of antibodies floating around for your protection. In your immune system, the cells truly are at work.